firstly, let me begin by thanking DeFi for the wonderful introduction, for organizing and orchestrating my entire agenda. I'd like to thank a few more people, NIHERST in particular, for inviting me here for a second time. First time to honor me with the award, and second time to talk to you guys. NIHERST is doing a phenomenal job in popularizing science and technology here, and I'm just happy to be a small part of it. Thank you so much for having me. I'm not much of a podium guy, so I'm just going to walk up and down, talk as much as I can. If you guys have any questions, you can ask me during the presentation, after the presentation, or you can contact me via email at any point. I'm easy to find, rkbirla@uh.edu. Before I actually, I typically just jump into these and talk to students and researchers about tissue engineering and organ fabrication. But this audience is a bit different. You guys are youths, very young, and my purpose here is to educate and inspire you to become the next generation scientists, the thought leaders, the people who are going to move the technology forward. So instead of jumping right into the science, PowerPoint slides never educate or inspire anybody. What inspires people are personal stories. So I'm going to just spend the first 5 minutes telling you a bit about who I am and how I got to where I did, so that you can see it's not as hard as it looks. I'd like to start by telling you I'm as Trini as can be. I was born and raised here. I spent the first 25 years of my life right here. I live in Houston. I drive in all these big highways. I don't care about them. Today, Michael was supposed to bring me here. I told Michael, forget about it. I'll find my way here. I drove around the streets of St. Augustine. I never get lost. I never run into any issues. And it feels so peaceful and great to come back home. I know my way around every corner. My background. So I went to University of the West Indies. I got my bachelor's in chemical engineering here. Actually, we can even take a step back. I went to, you guys are high school students. So I went to, not high school students, mainly A-level students. I went to Hillview College for my A-levels. Are there any? That answers my question, but to make it more official, how many Hillviewers? Okay. You would have been better off not raising your hand because I didn't like it there very much. Nothing personal. Nothing personal. Before that, I only went to Hillview College for A-levels. So I'm not much of a, whatever Hillviewers are called. My high school education was from Tunapuna Government Secondary School. And I know I don't think there's anybody from that school because is there anybody from TGSS? No, right? You have TGSS? Wonderful school. Love it there. So TGSS, Hillview College. Then I got my bachelor's degree in chemical engineering from the University of the West Indies. This is my home. This is my educational institution. One of my best professors, Professor Mallows, is here with me in the audience. After that, thank you so much for the education that you provided. It served me well. Then I went on to get a PhD in biomedical engineering and started research in artificial heart development. So basically, I'm just one of you guys. And if anybody has an interest in pursuing higher education, it's not as hard as you think it is. Back when I was looking for schools and institutions, we had a tough time gathering information and so forth. If you're passionate about science, follow your dreams, follow your heart. Any one of you and every one of you could be and should be standing in this podium within the next 10 to 15 years, and I'm sure you will be. With that background in mind, I want to sort of start into the research part of it. I have 1 hour. I don't have a clock, so when my 1 hour is over, just shut me down. My research is focused on tissue engineering. Tissue engineering and organ fabrication. This is a brand new field. The field itself was started about 10 years ago. When I entered into the field, there was nothing in the field, and nobody even knew what tissue engineering was. There was not even a definition of tissue engineering. That's still the case. It's still very fuzzy in terms of exactly how tissue engineering is defined. To keep things simple, I teach a lot in this area as well. What exactly is tissue engineering? tissue engineering equals organ fabrication. How do you bioengineer artificial organs in the lab? And when I talk about artificial hearts, my research is focused on artificial hearts, but this can apply to any tissue or organ system. People often think about the Jarvik hearts. The Jarvik hearts are these mechanical pumps. How many of you guys know about the Jarvik hearts? Okay. So the way it works is Jarvik hearts are mechanical pumps. They're pumps made of stainless steel, all these valves, all these mechanical components. Those have been used in patients. But the problem with these mechanical pumps is exactly that. They're mechanical. My heart is not mechanical, neither is yours. So our goal is to be able to replace these mechanical devices with completely biological devices. How can I bioengineer a completely bioartificial heart that's exactly matched in form and function to my heart and your heart and use that in patients? That's what artificial organ development is about. Functionally and structurally matched to mammalian hearts. I just want to get feedback on one thing. I'm focused on bioartificial heart development. If you had an open slate and you can bioengineer any tissue or any organ, I'm going to take 3 answers. Which one would it be and why? Raise your hand if you'd like to chip in. What would you like to bioengineer and why? And then I'm going to tell you how to do it. Go ahead, the person in red.
I would bioengineer skin because you have a lot of animals like whales and dolphins and stuff that get injured, and normally it's called skin only deep, but they can't heal us. They don't have medical, they don't have
They don't have the ability to regenerate the skin. They don't have the ability to, and that's a big, big problem when it comes down to burn victims. You need to be able to cover the skin as fast as possible. So wonderful answer, and actually some of the earlier work in tissue engineering was focused on skin. So you will do that very soon. Come on, let's hear something else. Guy in green. I can use one of those actually. Yeah, definitely. Yeah, good one. Go after it. Anybody else? So one more hand goes up. Person in blue. It is, and you will bioengineer a bioartificial lung. And just FYI, anytime I start teaching a class in tissue engineering, I always ask the same question, and everybody in the class says the heart. None of you guys said that. The heart is important, and you guys should engineer the heart. People in my class say that because they want to keep me happy. You guys don't care about that. That's fine. Okay, let's get right into it. So this is a long presentation, 120-something slides. I'm not going to go through all of those. We'll just touch on a few of the main elements. But when I give my typical tissue engineering presentation, here's what we cover. So introduction and overview to tissue engineering. What exactly is tissue engineering? How is it defined? And what is the tissue fabrication process? We spend about 10 or 15 minutes on that. If you don't get anything out of this lecture, at least I want you to have an appreciation of what tissue and organ fabrication is about. I think most of you guys are going to be here for the workshop tomorrow as well. Is that right? Everybody comes, I think. And tomorrow we will do some hands-on demonstration of what some of the principles of tissue and organ fabrication are. So if you need to take a nap, stay up for the first 10 to 15 minutes, and after that it's a good time to doze off. So we'll go over what is tissue engineering. After that, with that background in mind, our lab is focused on developing strategies and technologies to bioengineer parts of the heart or the entire heart. And I'm going to go through some of these models with you. With bioengineered artificial heart muscle, tissue-engineered vascular grafts, which are blood vessels, tissue-engineered heart pumps, bioengineered valves, ventricles, and entire bioartificial hearts. And I'll show you, firstly I'll start by going through the step-by-step process of what tissue engineering is all about, and then I'll illustrate these with a few examples from the cardiovascular space. Okay? Okay. Why? Why do we want to bioengineer bioartificial hearts? Organ transplantation is a major, major problem. In cases of chronic heart failure, the best strategy, the best treatment modality is for your cardiac surgeon to treat the patient by transplanting a heart. However, globally, the number of donor hearts available is always less than the number of patients that require heart transplantation. Thousands of patients die while on the wait list. In order to avoid that problem, instead of having patients wait on a wait list for an organ transplantation, we believe we can bioengineer bioartificial organs and make them available to patients when they need them. The whole strategy is, or the whole vision is off-the-shelf organs. That's what we're going after. Even for those that do receive heart transplants, there are problems associated with it. Those patients need immunosuppression therapy. The quality of life is suppressed, and the long-term life expectancy is still very low. If we can solve all of these problems by bioengineering bioartificial organs, we think it is a worthwhile goal to pursue. This is for the heart. It's across any tissue or organ systems. You will see the same problem. Okay. Formally, what exactly is tissue engineering? This is the formal definition for it. Tissue engineering is a multidisciplinary field bringing together experts from engineering, life sciences, and medicine. So that's one thing I wanted to point out. The multidisciplinary nature of tissue engineering and or any advanced research. Engineers, life scientists, and surgeons all have to work together in order to solve problems in medicine. For those of you who move forward to advanced degrees, who pursue graduate studies, undergraduate studies, always remember, no one person can solve any problem in science or medicine, and all research is collaborative, and you always have to work together with people from different disciplines. Tissue engineering is a multidisciplinary field bringing together experts from engineering, life sciences, and medicine, utilizing the building blocks of cells, biomaterials, and bioreactors. Cells, biomaterials, and bioreactors. Those are the fundamental elements of tissue engineering. Cells, biomaterials, and bioreactors. No matter which area of tissue engineering you're working in, you cannot get away from these. And tomorrow in the workshop, I'm going to illustrate this further. For the development of three-dimensional artificial tissue and organs which can be used to augment, repair, and or replace damaged or diseased tissue. This is the formal definition. Nobody ever remembers this except the students who get grades for it. As far as you guys go, tissue engineering equals artificial organ development. That's all you need to know. Okay? This is the figure that goes with the definition. Tissue engineering, building blocks of cells, biomaterials, and bioreactors. Participating disciplines, engineering, surgery, life sciences. And the application is tissue repair or replacement. This is the heart of tissue engineering. This is the 8-step process to bioengineer any tissue or organ systems. Anybody who has ever taken a class with me has to know this. If you don't remember anything from taking my class, you sort of have to remember this. I'm going to go through some of the main steps, and we'll talk about it. This is generic for any tissue system. It's not specific for the cardiovascular space. Step 1, cells. I can talk to you guys for hours about cells. What is a cell? The cell is a functional element of any tissue. In the case of the heart beats. Every time the heart beats, a single cell, known as the cardiac myocyte, contracts. It gets shorter, longer, shorter or longer. Each one of those contractions results in the heart beating. So what happens at a macroscopic scale is due to the result of what's happening at the microscopic scale using, as a result of this cell contraction. The biggest question you have to ask yourself in tissue engineering is, if you want to be able to bioengineer any tissue, where will the cells come from? That's a $100 million question, and if anybody has the answer to that, you're going to be rich on the spot. Where will the cells come from? That's a big area. That's such a big word. Which type of stem cells? Pig? You said pig stem cells? Okay. If you use pig stem cells, which you can, then you're going to need to put the patient on immune suppression, because then that's pig cells being transplanted into human cells. So autologous cell therapy is better. Patient-derived cells. So don't use pig cells. What else? Let's just spend 2 minutes talking about this. Each one of these squares is just such a big topic that I can talk to you for hours about each one of these. But I'll just pick one or two and talk to you for a minute or two. Cells. Where will the cells come from? Stem cells. When stem cells is in the news every day. When you hear stem cells, what do you think about? Anybody? Embryo? Yeah. Okay. Cloning. What else comes to mind? Moral dilemma. What else? What is the moral dilemma? Or destroying life. Right. Okay. Human embryonic stem cells is a very, very controversial area. One potential source of cells is human embryonic stem cells, which is cells that are derived from a human embryo. Those cells have the potential to become any cell type of the human body. But in the process of doing that, you have to destroy the embryo. In essence, you're taking life. And that is a big controversial area. People in general have moved away from that. About 10 years ago, people were looking into stem cells from an embryonic standpoint. So the ethical aspects diminishing over time. And now there's something called induced pluripotent stem cells. Actually, induced pluripotent stem cells means a skin cell. I can take a skin cell from any patient, take a skin cell, reverse engineer it to become embryonic stem cell, and then forward engineer that cell to become, say, a cardiac myocyte. So whatever the embryonic stem cell was doing, the skin cell can do now. So you do not have to destroy the embryo, and you have autologous patient-derived cells. Therefore, immune suppression is not an issue. Have you guys come across IPS cells, induced pluripotent stem cells? You guys know? Somebody said yes. Who said yes? Somebody. Okay, so that's where the new technology is going.
Sorry.
Yes. Question?
What couldn't you use the cell, the last, the deepest layer of the epidermis, that is the scaffold that is visible?
Can we use cells?
Yes.
We use skin-derived fibroblast, and there have been about a dozen different stem cells that have been tested to the basic concept of taking an adult-derived stem cells to form an embryonic stem cells and then forward engineer them has been proven in about 12 to 15 different cell types. So was there a specific cell type?
What I'm saying is that the growing layer of the epidermis will also be engineered to produce other cell types. That is the deepest layer of the epidermis. The scaffold that is visible.
Definitely. I'm sure they've tested that as well. The best one that I've looked at is adipose-derived stem cells. That's basically fat cells. Imagine if you can take your fat and convert that into bioartificial hearts. Who would not want that, right? Okay, that's enough about cells. Biomaterials. So what exactly is a biomaterial? If you want to build a house, you need some type of material. Anything you need to build, you need some type of a structural support. Mammalian tissue is made up of two components, cells and ECM. Cells provide the function. The ECM, which is your extracellular matrix, provides structure. When we bioengineer tissue, the cells are cells, we use cells, and in order to provide structure, we engineer materials that mimic the properties of mammalian tissue. Those are known as biomaterials. So how do you, just think about it. So I asked the question, where will the cells come from? Where will the material come from? What material are you going to use? Any thoughts? Mammalian-derived ECM, polymers, scaffolds, wood, concrete. What are you going to use? Any thoughts? Anybody has any? So that's where people started with. They have no idea what material to use. You have to bioengineer materials, starting with individual monomers. You have to go through a complex series of material processing steps in order to bioengineer the material so that it's similar in form and function to mammalian ECM. That by itself is a whole area of research. Genetic manipulation, you have the cells, you have the material. Before bioengineering three-dimensional artificial tissue, in most cases what we want to do is manipulate the cells in such a way so that they're more viable when we bioengineer the tissue. One example is we can increase the expression of contractile proteins so that they contract faster, generate more force, and they're more functional. Cells, materials, genetic manipulation, you combine all of this and you end up with a three-dimensional tissue which is somewhat similar in form and function, but it's not quite there. This is your first generation scaffold. It has some properties of mammalian tissue, but not all. Once we get to this point, the next step, I embedded sensors. Sensors need to be designed to test the functional properties of three-dimensional tissue. These sensors do not exist. We have to make the sensors ourselves. Sensors need to continuously measure oxygen, glucose, pH. You need to measure the calcium properties, the electrical properties, the contractile properties in a positive feedback loop. Once you have that in place, the next step are the bioreactors. What is a bioreactor? If you think about the heart is beating. It's mechanical, meaning every time it's beating, it's generating force. It has fluid stresses because the blood is flowing. It has changes in the electrical current because your heart beats in response to the depolarization wave, which is electrical. You have changes in the hormonal concentrations. You have changes in the growth factor. So there's so many things taking place in the heart. Electrical, mechanical, chemical. In order to mimic those conditions in the lab, we design custom bioreactors. These bioreactors are designed to emulate the properties in vitro, meaning whatever happens in the body happens in these bioreactors. And these are designed to support tissue maturation. After you do that, you need vascularization. Why do you need vascularization? Why do we have vascularization? What does our vasculature do? Our vasculature supplies oxygen to all parts of the body. Without a vasculature, oxygen can only be transported from one cell to another cell through a thickness of 200 microns. If you're bioengineering tissue, which is more than 200 microns, the center core of the tissue will necrose. Once you're above 200 microns, you need to be able to figure out how to bioengineer a vasculature within the three-dimensional tissue. And once you've gone through all of these steps, then you need to test your three-dimensional tissue construct both in vivo and in vitro before you can use it for patients. Okay? In a nutshell, does that make sense? Okay? Okay. Complete sense? Some sense is fine. Let's move on. This is just a description of everything. Okay. How are we doing for time? I can see that Dr. Fellows will watch, so I'm fine. Okay. Okay, I'm not going to go into all of this. Heart anatomy. We are not going to go into anatomy. This is what your muscle looks like. This is what your blood vessel looks like. These are what valves look like. These are what pumps look like. These are what ventricle looks like. These are what whole hearts look like. I'm not going to explain the anatomy. I just want to illustrate one thing. These are what our three-dimensional tissue construct look like. This is the 30,000 feet view. I just want you to sort of zone into it. These are bioengineered constructs, and I just want you to have an idea of what it looks like, and I'm going to go through as much as I can within the next 30 minutes or so. Okay? Okay, three-dimensional artificial heart muscle. I'm actually going to get into some of the technical details now. If this doesn't make sense, feel free to ask me, stop me and ask me. How do you bioengineer three-dimensional artificial heart muscle? We have developed at least 6 different platform technologies to bioengineer three-dimensional artificial heart muscle, 2 of which are based on scaffold-free technology, 1 which is based on polymeric scaffolds, 1 on biodegradable hydrogels, 1 on acellular scaffolds, and 1 which is completely developed in vivo. These are completely different methods to fabricate artificial heart muscle. At this stage, we do not know which one is better. Each one has advantages and each one has disadvantages. If you build a house, you know exactly what goes first, what goes second, you know exactly what to do. That's not the case with this. There are many different variables involved, and we haven't figured out which one is the best. Okay, let's look at scaffold-free technology. So one example. So let's get into some of the technical details. We start, I'm just going to go with it. We start with a 35 millimeter tissue culture plate, which has been pre-engineered with anchor points. The plate is coated with PDMS, followed by laminin. Once the surface has been specifically engineered, primary cardiac myocytes are isolated from neonatal rodent hearts and plated at confluence. Once these cells are plated at confluence, they form a sheet or a monolayer of cells on the surface. Over time, these cells are contracting. As the cells contract, they pull and cause the sheet to detach from the outside layer. This sheet moves radially inwards towards the center of the plate. At the center, it sort of wraps around and forms three-dimensional artificial heart muscle at the center. This is a movie. I think I have to go up to click it. Okay, so this is an example of one step of the formation. What you have here is a sheet or a monolayer of artificial heart muscle. This is beating in synchrony. It is beating in unison. There's nothing coming in the way. It's just all one big sheet of artificial heart muscle, which is beating and doing what it's supposed to do, similar to what your heart muscle and my heart muscle should be doing. It's one step in the process, and at the end of it all, you end up with a strip of heart muscle that looks like this. Okay. The white stuff in the middle is a skinny little strip of artificial heart muscle. There's three-dimensionality associated with it. This was fabricated 10 years ago. So it's very small, it's very weak. It cannot be used to support heart function for a person or even for an animal. The functional performance of this guy was 70 micronewtons. It needs to be at least 10 times more, and we're about halfway there. But this was one of the first bioengineered three-dimensional tissue constructs that was fabricated in our lab. What is the big deal with this model? These are some of the functional performance metrics. The big deal with this model was it's based on self-organization strategy. What does that mean? I told you that the most important components of bioengineered three-dimensional heart muscle are cells and biomaterials. This was bioengineered in the absence of any biomaterials, meaning the cells do all the work. Why is that important? That's important because the perfect biomaterial has not been fabricated as yet. If we can trick the cells to making their own ECM before forming three-dimensional tissue constructs, they will do a better job at it than we can do by engineering any material in the lab. So that's this whole area called self-organization technology. Okay? Let's just go through a few more of these models. I'm going to show you one more. Let's not get into all the details. This one. Okay. So what's happening here? This is a big ball of three-dimensional artificial heart muscle. This guy is magnetically charged. Again, this is based on self-organization technology. What does that mean? No ECM. So what we do is we take cells we mix some magnetic nanoparticles with the cells. So in essence, what we have done is we have magnetized cells. If you have magnetic cells, you can then you can use magnetic fields to cause the cells to clump together, come together, form these aggregates. That's exactly what's happening here. These are magnetically guided tissue fabrication strategies. No ECM required, magnetically charged. The magnetic charge is under your control. It has no adverse effects on cell behavior. And then you can use controlled, guided magnetic fields to determine where the cells go, cause them to come together, and beat. Some of these models somewhat making sense. Okay. Do you have any specific questions? I don't know. I've gotten into some of the technicalities of it. I'm going to go through a few more models, but is this somewhat making sense? If it's not making sense, you at least, you can see the beating. That's what we're going after, artificial heart muscle. Any specific questions at this point?
Yeah.
Please.
I understand that you are focusing on modeling the cardiomyocytes.
Right.
But we know that for the functional heart, there are other cells that participate in the applicability of these cells. There are also the nodal cells,
yeah,
which enable the heart to control itself.
Right.
And there are also nerves coming to modify that result.
Yeah, that's absolutely right.
Factoring all those into the
Yeah, yeah, that's very important, and we are factoring all of those cells in. So these cells are engineered with a specific concentration of myocytes, specific concentration of fibroblasts, specific concentration of SA node cells. All the cells are in there. We don't know exactly what the right proportion of each one of the cells are, but a lot of the other research in the area, for example, if you read some of the earlier work, people would just take the cardiac myocytes and they would call all the other cells contaminants, bad. But what we have come to realize is what you said, that all the cells need to be in there, otherwise it's not going to beat. It's not going to have the fibroblast in there. It's not going to generate ECM. Okay?
Now, the magnetic union between the cells, put them together, would it function as the interconnected disk of the normal heart?
It doesn't. The magnetic particles are in the range of microns. They're very, very small. The magnetic fields are very, very weak. They have, based on our tests and others, they have no influence, no negative influence on the cell behavior. They have no negative influence on biocompatibility, meaning they don't damage the cells, they don't kill the cells. All they do is they provide us with a mechanism to guide cellular positioning. So the whole magnetically guided tissue fabrication is something very new, and it's turning out to be a very powerful tool at our disposal. And it doesn't come in the way of the contractions as well. Okay. Now these are some more models. I'm just going to show you a few more contractions before we move on to this. Biodegradable hydrogels. Let's talk a bit about biodegradable hydrogels. So what exactly is a biodegradable hydrogel? The most important component, or the most important structural component of mammalian heart muscle is collagen. Collagen is a structural protein which provides structural support to the heart muscle. Biodegradable hydrogels, they're very functional, meaning they have cell attachment points. They interface very well with cardiac myocytes and have been proven to be a very effective tool in bioengineering artificial heart muscle. In this case, we don't use collagen. Collagen is hard to get. We use fibrin. Fibrin can be obtained from blood, easily accessible. And what we find is if we create a layer of fibrin first, anyhow, I'll go over the technology. What we have is we start with a tissue culture plate, and what we do is we create this coating of fibrin first. So the fibrin goes first, which is, over here. Once you have the fibrin, then you add the cells either on top of the fibrin, or you can mix the cells with the fibrin. So in the previous cases, we were using self-organization strategies, but in this case, we're using fibrin to give the cells something to grab onto, give the cells something to attach to. If we do that, a lot of the technology is the same, but at the end of it all, you end up with a three-dimensional patch, which looks like this. Let's get into I have some videos here. Okay. So this here is a 2 centimeter, can you guys see it is the lighting okay? Just where I'm seeing it from is not that clean. Okay? So what we have is a 2 centimeter by a 2 centimeter patch. This has been sized for a rat heart, and it's beating very violently. It's beating very rigorously, generates very high forces in the order of millinewtons. When I was talking about the self-organization strategies, which was developed 10 years ago, those are generating 70 to 100 micronewtons of force. These guys, 10 years later, this work was published in 2014. The technology development cycle took us 10 years. 10 years later, we were able to bioengineer artificial heart muscle, which was generating 6 to 8 millinewtons of force. Very, very high forces. And this is based on the fibrin gel model. Okay? These are the depolarization waves. You have coupled electrical stimulation, which leads to contraction of the heart. So these are your depolarization waves. These are your electrical conduction waves. Everything is very well synchronized. What this means is, for every depolarization wave here, we have a 1-to-1 alignment, meaning every time your heart fires a depolarization wave, it's generating contractile force. And we have very good control over this. Okay? Electrical pacing, your heart needs to go continuously. These guys are able to go at 1, 2, 3, 4, 5, 6, 7 hertz. If you look at the histological characteristics, what does histological characteristics mean? Meaning if you chop it open and you look at what's happening inside, everything looks the way heart muscle does. If you look at these images in particular, all the green stuff here are your contractile cells. The contractile cells are your myosin heavy chain. Those are the ones, every time the heart beats, the myosin heavy chain and the alpha actinin generate twitch force. All the green stuff here are the contractile proteins. All the blue stuff here are your cell nuclei. So what we found is when you chop these guys up and you look at them, there's a lot of contractile protein here. If you look at the striation patterns are similar to what you would expect from mammalian heart muscle. Anyhow, let's not get too much into all these technical details. And since we're working in three-dimensional space, I just have this one video which shows that we... anyhow, let's not worry about that, okay? These are some of the metrics we use, EKG monitoring. I mean, we don't have to get into all of these tools, okay? I think rather than going into all of these details for the different heart muscle models, I'm actually going to move on to some of our other tissue systems, okay? Okay. This is also a three-dimensional heart muscle that was bioengineered. All the ones that I've showed you so far were completely bioengineered in the lab using cell and tissue culture techniques. This one was completely bioengineered in vivo using an animal model, meaning we inject the cells, we inject the matrix, everything happens inside of the body. This one turned out to be bigger, better, faster, stronger compared to the in vitro models. Why is that? Because if you can do everything in vivo, the in vivo environment is much, much better than the in vitro environment because you have all the right physiological cues. Okay? Movies are stalling a bit. Okay. Okay. I want to talk a little bit about the bioreactors. I want to show you a few things. Okay. So it's one thing to be able to bioengineer three-dimensional heart muscle. The other part of it is bioreactors. We need the bioreactors for mechanical guidance, for electrical guidance, for perfusion, for electromechanical stimulation. Why do we need these bioreactors? We need bioreactors to be able to simulate in vitro conditions. We need to be able to stimulate in vivo conditions in vitro. We need to be able to recreate all of these cues that happens in the body. We need to be able to recreate those in the lab. Let's see if this one plays, okay? Do you see this strip of muscle here? This is three-dimensional artificial heart muscle. This is all the instrumentation that goes into applying a controlled stick-lick stretch. Meaning you have to sort of grab onto the three-dimensional artificial heart muscle here, If you look carefully, it's contracting here as well. Videos are not playing more than once. Okay. Let's think. Here's another one. So this is a three-dimensional cardiac patch. These are custom bioreactors which are grabbing onto the three-dimensional artificial heart muscle, pulling on it. If you give it a cyclic stretch, a uniaxial cyclic stretch, what happens is that the three-dimensional artificial heart muscle becomes bigger, better, faster, stronger. It's a better muscle altogether. The instrumentation that comes for it, you need to be able to build the instrumentation. You need to be able to develop specific protocols to be able to deliver uniaxial stress. Okay? I think that's enough about heart muscle. I'm going into some of the other models here. Tissue engineered vascular grafts. How much more time? About 15 minutes or 15 minutes? 20 minutes? I'm still okay for time. Let's go into tissue engineered heart pumps, okay? Okay, let's talk about tissue engineered heart pumps. What exactly is a tissue engineered heart pump? How many of you guys know what an LVAD is, a left ventricular assist device? Anybody heard about that term? Left ventricular assist device, LVADs. Okay, anybody? In the what? Grey's Anatomy. That's good, you heard about it. Anybody else? Okay, let me tell you what an LVAD is. An LVAD is a left ventricular assist device. It's a mechanical pump. Say a patient comes in with chronic heart failure and he or she needs a heart transplant. The patient is put onto a wait list. If a heart is available, the patient gets the heart transplant. If a donor heart is not available, then what your surgeon can do is they can attach this pump, which is a mechanical pump, and it's used as a bridge to transplantation. While you're waiting for a heart transplant, the left ventricular assist device can be used to support your heart function. It's designed to provide temporary relief. By temporary, it's about in the order of 1 year to 2 years until you can find the donor heart. What exactly does it do? It's a pump that connects directly to your left ventricle and takes over the job of the left ventricle and pumps blood directly from the left ventricle all the way to the aorta. It basically takes over the function of the heart. It's a mechanical pump. In order for your surgeon to implant that in you, it's an open heart surgery. They need to drill a hole in the apex of the heart, put that in, spend 1 hour stitching it on, and after that, you have a lot of issues. You have a lot of blood clotting. You have a lot of immunosuppression. You need to be on immunosuppression, and the pumps do fail because they're mechanical devices. One way of overcoming these problems is, can we bioengineer cell-based devices or cell-based pumps that can accomplish that same objective? So when we're talking about heart muscle, you're talking about something that exhibits contractile force. And the most important metric is twitch force. How much force does it generate? When you're talking about pumps, you're thinking about a linear tube that's generating pressure. You know, if you have a tube that's generating pressure, and that can be used to move blood along the direction of the blood flow. So that's the basic concept of cell-based cardiac pumps, and that's what this is about. So this is the basic concept here. These are the devices. If you have something that can generate force, you can use this if it contracts, you can have blood flow through this guy. Endothelialization, valves, these are the bioreactors that I was talking about, the electrical stimulation, mechanical stimulation, and these are functions to work. So this is the left ventricle. These devices are attached directly to the left ventricle and are used to pump blood directly from the left ventricle to the aorta, completely bypassing the need for the left ventricle to function. Okay, here's an example of a cell-based biological pump. This was made using decellularized blood vessels with a sheet of heart muscle wrapped around it. It's beating, but instead of beating like the sheets were beating, it's doing this instead. If you put a pressure transducer in here, you can measure the twitch pressure, not the twitch pressure, interluminal pressure, which is in the order of 10 millimeters of mercury. A lot less than we need to be at, but it's still able to generate measurable pressures. The material we use here is a decellularized vessel. Decellularization technology means that you strip the cells out of mammalian tissue, and what you're left with is intact ECM, which can be used to support artificial tissue. Okay? This shows that what you have here is a big tube with all the blue stuff around here, all the cells. This is the histological data. What you do is you chop it up to see what it looks like from the inside. And what you have is these big tubes with cells wrapped around it. Okay? All the red, all the yellow stuff, again, this is confocal data, which is used to look inside the three-dimensional tissue. You can see it's contracting, but what does it look like inside? Here is all of your ECM components, intact, structural. This is all collagen, meaning it's rich in collagen. Collagen is required to provide structural support. All the blue stuff are your cells on the outside. If you have cells on the outside and collagen on the inside, it is going to generate interluminal pressure. Okay? These are all the functional properties. It generates pressure. You can pace it at different frequencies. It has all of the conduction velocities, the conduction maps, all the good stuff. Okay? I'm not going to talk about valves. I'm going to talk about ventricles and hearts. That should be, and I should be good for time. Okay? Okay. So we're getting into we're talking about tissue, but we're moving more towards the organ models at this point. How can you bioengineer a complete bioartificial ventricle? Why do you want to bioengineer a complete bioartificial ventricle, and how will you use that? There's some cases, a congenital disorder, HL, hypoplastic left heart syndrome. In this case, when a baby is born, it is born with an underdeveloped left ventricle that does not have the capacity to provide functional support to support the baby, the circulatory system. When I was hired at the University of Michigan, I was hired by a professor called Professor Edward Beauvais, who is one of the most renowned cardiac surgeons in this area. Here's how he explained it to me. We have so many of these kids that are born with hypoplastic left heart syndrome. What we need to do as cardiac surgeons is go into the heart. What they do is they open the heart, open the chest. They have to conduct various very invasive surgeries. And what they do is they re-engineer the underdeveloped heart to function so that it can support the heart. They re-engineer all the vessels. They put grafts in. It's very invasive, requires multiple surgeries, and the kid has to go through a lot of trauma. What he told me is, can you bioengineer a complete bioartificial ventricle so that when these kids are born, they can diagnose this during pregnancy. So if this is diagnosed during pregnancy, you have a few months before the child is born. Can you bioengineer a complete bioartificial ventricle? By the time the kid is born, we have the ventricle ready, and then we implant it into the patient. That's the vision. This was back about 10 years ago when I was at Michigan. We didn't get the project going then. I'm not going to get into all the details, but I'm going to show you what one of our ventricles looks like. Within the last 3 to 6 months, we've been able to successfully bioengineer a complete bioartificial ventricle, and this is what it looks like. So what is this? It's just a cone. It's in the shape of a ventricle. This is what your ventricle will look like with the right dimensions. That's one part of the ventricle. The second part are the actual valves. The valves regulate the flow of blood and control, it provides direction control of blood flow. So what we do is we engineer these ventricles separately, we engineer the valves separately, and we put this together to bioengineer a complete bioartificial ventricle. To the best of my knowledge, this is the only bioartificial ventricle ever engineered in the world. And we're about to publish this within the next 6 months. So it's very exciting for us. I promised my department, this is when I was at Michigan, I promised my department chair that I will bioengineer a bioartificial ventricle, and 10 years later, we have it in the lab. Once this work is published, he's going to be the first to receive a copy of that manuscript. I'm not getting into all the details, but at least visually, you can see that it looks like a ventricle. Visually, I also want you to think about the transition. I spoke a lot about those, those skinny little strips which don't generate a lot of force. Then we went into those planar models, which are sheets, which were generating much, much higher forces. Then we went into the tubes. You see the sort of increase in the degree of complexity. Now we are talking about complete bioartificial ventricles. How do we go from this to complete bioartificial hearts? So I spent the last 5 minutes talking about bioartificial hearts. Okay? Sterilization, bioreactors, all the good stuff goes in. This is just finishing off the slides for the ventricles. I don't want to go into all of this. This is what it looks like. This is how we cellularize it. These are how the bioreactors look like. This is what they look like in real life, and it really does exist. Bioartificial hearts. This is the crown jewel of our lab. How can we bioengineer bioartificial hearts? And before you leave here, I'm going to show you actual videos of hearts that are beating, that were generated in our lab. Okay. Concepts, you're sold on the idea that we need to be able to bioengineer total bioartificial hearts. It's important, and we have to do it. How do we do it? Too much details, I guess. I don't want to go into all of that. I'm going to skip a few of the technical details and just run into the heart of the project. So let's get back into the material side. We use something called Desalination technology. I alluded to it when I was talking about pumps, but I didn't explain it because I was going to explain it here. Let me just go over what this slide is showing. This is a freshly isolated, was there a question? Okay. This is a freshly isolated rat heart. So I pull this out of a rat heart, nice and red. I go through a series of decellularization technologies, and after 14 days, this looks significantly different from that. Okay? This is nice, red, because it has all the cells in it. After 14 days of decellularization, this looks translucent. It is white. What I've done is I've completely removed all the cells from this heart. When I remove all the cells, what is left behind is the intact extracellular matrix. Remember I told you tissue is made up of two components, cells and ECM. So what I've done is I've taken tissue, I've gotten rid of all the cells, and I'm left behind with an ECM. I then use this ECM to recellularize it, repopulate it with cells, and use this to bioengineer bioartificial hearts. Just off the top of your head, what's the big deal with decellularization technology? I'm not making this in the lab. What I'm doing is I'm taking something that has been engineered by nature. I'm taking a heart, using the ECM to support BH, bioartificial heart fabrication. Thought process is anything that was engineered by millions of years of evolution is going to be the best biomaterial out there. That's one part of the equation. Second part of it, from a clinical standpoint, how exactly are you going to get a heart to decellularize? You're going to take the patient's heart out, let the patient die, decellularize it, bioengineer the heart and put it back in the patient? No, right? You can't do that. This makes absolutely no clinical sense. So you cannot do this clinically. The reason we use decellularization technology is that we have no biomaterial that we can engineer in the lab that is even close to this material. So we view this as our positive control. First, we get this to work. And once we get this to work, we have all the sterilization technologies worked out. We have all of the bioreactor protocols worked out. In the meantime, we hope to be as good, or at least getting equivalent to be able to bioengineer these materials in the lab. So that's one part of the equation. Sterilization, the biomaterials. How do you cellularize this? Okay. There are two cellularization strategies. It's a two-stage cellularization strategy. One stage involves these artificial heart muscles that we make in the lab. You have your heart. We take these muscles. We wrap layers and layers and layers around it. These three-dimensional artificial heart muscles are contracting. As they contract against the decellularized material and generate intraluminal pressure. That's what this image is showing. You have a heart, you have a patch, the heart, you take the patch, you wrap it around, and you end up with a bioartificial heart. I'm going to show you a movie of how these hearts work. You see what's going on there? This here is your decellularized matrix. This here is your artificial heart muscle, which is contracting against this area. And as this contracts, it's generating intraluminal pressure, and the lumen will be right here. This generates some pressure, okay? So it's good. You can see it contracting. You believe it. What's the problem with this? Any thoughts on it? How do you attach it? That's one problem. Anything else? I hear a lot of talking. Is it because you're getting bored, or anybody has any ideas? How will it infuse? How will it integrate? How will it attach? All of those good things. How many layers do you use? But all of those are important problems. Let me tell you the biggest problem with this. You have something contracting here. You have nothing here. You have no cells. All you have is an empty ECM. That's not going to work. It's contracting against nothing. So the second stage of the decellularization strategy is, how do I get cells in here? It's a nice image, but this is what I wanted to show you guys. This is the best of what we have in the lab right now. What's happening here? What is this? Entire bioartificial heart contracting, sitting in a dish, beating by itself. This was done using, I told you that we needed to get the cells on the inside and then the patch on the outside. Initially, I showed you the patch on the outside. That works well. That's contracting. This one is only the cells on the inside. Doesn't have the patch on the outside as yet. This is a complete bioartificial heart that's contracting all by itself in the lab, only using direct cell injection. So far, we've done it with direct cell injection, and we've done it with the patches. They all generate very low pressures in the order of 2 to 3 millimeters of mercury. The next step is to combine these two technologies together and increase the intraluminal pressure. So that's the novelty, the two-stage cellularization process. Right now, we have done the both separately, and within the next couple of months, it is our objective to bring these two technologies together, and it is our expectation that the force is going to go, the pressures are going to go up significantly. Bioartificial ventricles are brand new. We just bioengineered them 6 months ago. Bioartificial hearts are even newer. This presentation was actually updated 2 weeks before I left, and that's when this movie was made. And there's nothing out there in terms of bioartificial hearts. So this is just brand new stuff coming out of our lab. And this is all the histological data. It looks good. All the green is your muscle. These are all the twitch forces, all the control hearts. It has all the EKGs. It has all the pressures, all the good stuff, all the bioreactors. Everything is in there. think that's it in terms of platform technologies from our lab. Before I get into acknowledgement, the two things that I want you to, I talked a lot about a lot of specific details, and probably I lost a lot of you guys in all the technical details, but don't worry about that. If the two things that I want you to remember when you leave here is, one, I want you to have an appreciation of the tissue fabrication process. Remember I showed you that 8-step process to fabricate bioartificial tissue? So keep that in mind, the 8-step process. If you don't remember anything from what I said today, remember the 8-step process. Also keep in mind, out of those 8 steps, how many steps did I actually talk about? All I spoke about was cells, biomaterials, and bioreactors. That's how young the field is. You're just beginning to understand the core technologies that go into it. There's very little out there in terms of sensors. There's very little out there in terms of vascularization. So the one thing I want you to remember, two things. One is the 8-step process. Second thing that I want you to keep in mind is I want you to have an appreciation of that complete linear sequence starting from three-dimensional artificial heart muscle, flat, sitting in a dish, contracting, all the way to those bioartificial hearts, which were entire hearts that were contracting in a petri dish. And think about that entire paradigm, and think about the entire transition and how much has been done so far and how much more needs to be done. With that, I'd like to acknowledge everybody who has been involved in this research. I'm not going to read out all the names, but I do want to say, mention the names of my mentors, Professor Miten Akai, Professor Dennis, Professor Edward Beauvais, Professor Frank Pagani, and Mike Welch, who have been instrumental in getting me to the point where I can present this research to you. You guys have been a great, awake audience. Thank you for your time and attention. If this makes some sense to you, I've done my job. If you have questions, you can ask me now. You can just look me up, rkberla@uh.edu, and email me. And I'm going to be seeing you for the workshop tomorrow as well. You can bring your questions to the workshop. I'm going to end it there.